So my name is Allison Wolfe, and I'm a PhD candidate at the University of Utah. And so my talk today is about people, mammoths, and grouse, and what all these have in common with case studies on the application of paleozoology to modern conservation. So my research centers around reconstructing past relationships between animals, the environment, and humans. And to do this, I've studied paleozoological records, and that can include paleontological or archaeological sites. And I link these to other data sets indicative of past environments and human behavior in order to investigate how different changes in environment or human impacts affected animal populations through time. And this work is important for modern conservation because modern wildlife conservation strategies are usually based only on historic records, which are records that span only 200 years. So the paleozoological records that I look at often span thousands of years or longer. And so they cover a wider range of conditions and thus offer the potential to greatly improve modern management, modern conservation and management as ongoing climate change and human impacts on the environment increase. So today I want to talk to you about two different case studies of my work that exemplify this approach. And the first one involves these guys. So at the end of the Pleistocene, 38 genera of mostly large mammals went extinct in North America. And this includes things like mammoths and mastodons, horses, and giant sloths. And in fact, over 70% of the continent's megafauna went extinct at this time. And the ecological consequences of these losses are significant. Such a dramatic reduction in large mammal biomass has significant effects on ecosystem function, including changes in biogeochemical cycling, changes in habitat and trophic structures, vegetation community composition, and even regional and global climate change. And today we're experiencing similar reductions and extinctions of large mammals due to factors such as climate change and human hunting and habitat loss. So understanding the causes and consequences of these losses in the past can thus have practical insights and applications for forecasting changes in or even restoring large mammal populations. Now, what exactly caused these extinctions is still debated, and in fact, it has been debated for over 2 centuries, and in fact, it's still one of the most contentious issues in North American archaeology today. And explanations include human hunting, climate change, or even disease or extraterrestrial impacts. But arguably the most influential explanation is known as Pleistocene overkill. And this is the hypothesis put forward by Paul S. Martin in 1967. So Martin noted that the timing of many megafaunal extinctions coincided with the earliest evidence of human arrival to North America. So he argued that once humans arrived in North America, they found a plethora of large animals that had no innate fear or defenses against them. So they moved quickly across the landscape, wiping off megafaunal populations as they went. Now, he did note that there were some evidence of climatic shifts that occurred at the same time, but he argued that climatic causes could not be the reason for these extinctions because these taxa had already experienced multiple interglacial periods, and these extinctions seemed to be focused on the large herbivores and the carnivores that depended on them, rather than the large suite of taxa that would be expected with a climatic cause. So he argued that when human hunters arrived in North America, it was the subsequent rapid, widespread hunting of these animals that led to their extinction. And this overkill hypothesis really caught on, and it's actually still popular today in popular science and in some areas of the scientific community, especially among ecologists. However, overkill has been subjected to intense scrutiny, and the most widely cited and empirically based objection is called the associational critique. The associational critique centers around the observation that very few sites contain secure associations between humans and extinct mammals. In fact, a recent assessment suggests that only 15 sites in North America have compelling evidence for such an association of humans and extinct megafauna. And they only include 5 of the extinct taxa: mammoth, mastodon, horse, camel, and gomphothere. If intense human predation was the cause of these extinctions, then, according to this critique, we should see more evidence of it. Among the longest standing supporters of the associational critique are Don Grayson and Dave Meltzer. And Grayson and Meltzer have argued for almost 3 decades that many extinct taxa were among the most abundant on the Pleistocene landscape, as evidenced by their abundance in the paleontological record, yet they occur less frequently in archaeological sites compared to extant taxa, the taxa that survived the end of the Pleistocene. They argue that this pattern is not consistent with overkill because it suggests that despite the plethora of large mammals that were on the landscape, humans weren't hunting them in any appreciable numbers. In their most recent iteration of this argument, so they plotted the number of archaeological kill sites against the number of fossil occurrences, or their paleontological abundance, for the 8 extant, so the taxa that survived the end of the Pleistocene, and the 5 extinct taxa that have thus far been found in secure archaeological kill or scavenging sites. And so the extant taxa include muskox, moose, elk, caribou, mountain sheep, pronghorn, deer, and bison. And the extinct taxa include gomphothere, camel, mammal, mastodon, and horse. And they noted that these 2 groups of taxa exhibit significantly different trends. So extinct taxa occur far more often in paleontological sites than extant taxa, and extant taxa occur far more often in archaeological sites than extinct taxa. So in essence, they argue that extinct taxa are underrepresented in archaeological sites compared to extant taxa. So this cannot support overkill. And so it is this visual representation of their recurring argument that they've been putting forth for decades that got my collaborator Jack Broughton and I thinking, because we were not convinced that there should be 2 separate trends for these 2 groups of taxa. And so we figured that because these data fundamentally address decisions that human hunters are making, which taxa they should hunt and which they shouldn't, we thought that examining it in a theoretical framework that addresses foraging decisions would be useful. So we took this idea of their analysis. We broadly agreed with their approach, but we wanted to modify it. So we looked at it through a foraging theory perspective. We used the prey choice model specifically, and we re-examined this data. And so this started kind of a back and forth between us and Grayson and Meltzer over a series of papers. So I'll just present to you the synthesis of that debate, and then I can answer questions about the details of it at the end if there are any. So looking at Grayson and Meltzer's data at face value, we came up with 2 hypotheses for why this apparent low number of kill or scavenging sites for extinct taxa relative to extant taxa might exist. So it might be that hunters bypassed would-be extinct large mammal genera on encounter. In other words, they just weren't hunting them, but they were hunting extant taxa. These taxa were excluded from their diet for whatever reason. Or it could be that those taxa that would ultimately go extinct were rarely encountered by humans, so they were not hunted as often. So this first hypothesis seemed unlikely to us, given the implications of the prey choice model. Now, I know many of you are familiar with this, but stick with me. So the prey choice model attends to conditions that influence whether or not a given prey type should be pursued upon encounter. So according to this model, human hunters aim to optimize their overall return rates by pursuing only an optimal set of prey. So high-ranked prey, which are those prey that have the highest post-encounter return rates out of all the prey items in a patch, should be hunted whenever encountered, and lower ranked resources should only be added as encounter rates with those high ranked resources decrease. Now, there have been many empirically derived return rate estimates along with ethnographic and experimental cases that have documented a significant positive relationship between body size and return rate. So bigger bodied animals are higher ranked. Now, there are some important exceptions to this, but they notably occur in contexts that don't have ungulates. So in this context with megafaunal taxa, others, notably Eugene and Byers, have concluded that all taxa as large or larger than a mule deer represent high-ranked resources. So we argue that all 13 of the taxa examined here would have been high-ranked, and thus humans should have hunted all 13 taxa, both those that would become extinct and those that would not. So for this reason, we reject this first hypothesis. The second hypothesis, that extinct taxa are underrepresented in archaeological assemblages because they were rarely encountered by human hunters, seems more plausible. So again, operating with that logic that all of these taxa are high ranked, they should have all been taken upon encounter. So for all 13 of these taxa, the more abundant they were on the landscape, the more they would have been encountered, and thus the more they would have been hunted. This suggests a positive relationship should exist between a taxon's abundance on the landscape, as measured by their abundance in the paleontological record, and its archaeological abundance. So the key to testing this hypothesis is to assess the abundances of these animals during the period when Paleo-Indians could have encountered them. To this end, we note that Grayson and Meltzer use two different time scales for their axes. So they have the Clovis period for the number of archaeological kill sites, which in this case they're defining as 11,500 to 10,800 years ago. But for the paleontological sites, they're including the entire late Wisconsin, which goes back 35,000 to 10,000 years. So it could be that this overrepresentation of extinct taxa in the paleontological record relative to extant taxa has to do with the fact that they were more abundant earlier in the Pleistocene, but declined in abundance by the time human hunters were on the landscape. So this alone could account for this difference in trends that Grayson and Meltzer advocate for. So we reason that an analysis focused only on the period after human arrival would not reveal such differences between extant and extinct taxa, and should show a single positive relationship between paleontological and archaeological occurrences for all 13 taxa. And this pattern would refute the associational critique because the number of archaeological sites would not be a result of hunting intensity, it would be a result of their abundance on the landscape. So to test this, we needed to make sure that all of the paleontological and archaeological records that we were using came from the same time period in the same geographic location. So we only wanted to look at sites that occurred during the overlap of humans and megafaunal taxa, and then we limited the geographic location to the contiguous US. And so once we gathered these data, made sure they matched in time and space, we plotted them in the same way that Grayson and Meltzer did, and we plotted the trend lines for extant and extinct separately, and you can see that they have nearly the same slope. So for all 13 of these taxa, the number of archaeological sites is scaled by their number of paleontological sites, which is what we expected with our prey model logic. So that means that extinct taxa are not underrepresented in the archaeological record, as the associational critique would argue. And we also fit a generalized linear model to all taxa together, and it showed that, as predicted, there is a single positive trend between archaeological abundance and paleontological abundance for all taxa, extant and extinct. And in fact, the model fit shows that 69% of the variation in archaeological occurrences is accounted for by variation in paleontological occurrences. But what about the other 33 genera? I mentioned at the beginning that 38 genera of mammal taxa became extinct at the end of the Pleistocene, and so far I've only talked about 5 that have showed up in archaeological context. So there are 33 other genera that have yet to be found in archaeological killer scavenging contexts. Many of these have last appearance dates before the arrival of humans, and so they're not expected to have ever had contact with humans. Humans likely never encountered them, but that possibility cannot be ruled out. So the relationship between archaeological and paleontological abundance that we identified here and modeled with the generalized linear model can actually be used to test how many archaeological occurrences we would expect for each of these 33 taxa based on their paleontological abundance. So we gathered the paleontological abundance for each of these 33 taxa during that period of overlap in that geographic location, and we inputted them into our generalized linear model. And so this is just the same model in plot as before, but down here, these triangles, those are the other 33 taxa. And so a lot of them overlap, which is why there's not 33. But this horizontal line here shows one archaeological occurrence. So when we input their paleontological occurrence into the model, for every single one of these 33 taxa, they all fall below one archaeological occurrence. So we should not expect to ever find them in any archaeological sites. And so that is consistent with the suggestion that the absence in the archaeological record of these other 33 taxa is due to the fact that they were either extinct or exceedingly rare on the landscape by the time humans arrived. So their absence and the presence of these other taxa in the archaeological abundance is predicted by prey model logic. So to conclude, our analysis shows that the associational critique, which is the most widely cited and widely accepted critique of overkill, is not valid. There are not too few archaeological sites containing extinct megafauna. There's just as many as we would expect based on their abundance in the paleontological record. But this doesn't support overkill either. It just shows that people were hunting these taxa in the, it shows that people were hunting taxa in the proportions that we would expect. But whether or not this hunting led to the extinction of any or all of these taxa remains to be seen. But the bottom line is that we cannot assess the cause of extinctions with these data alone. So while our analysis did not lead us to a clear cause of extinction, it was instructive in that it showed us where we need to go in order to get to the bottom of the extinction debate. So instead of these continent-wide assessments of abundance data, future analyses need to focus on other data sets, such as paleoenvironmental records, demographic histories, and ancient DNA at scales involving individual taxa at smaller geographic regions. And a number of researchers are taking this on, and it seems like the vast majority of them are finding that a climatic cause is more consistent, or these studies are consistent with a climatic cause for these extinctions rather than overkill. So just to give you an idea of this, this is a figure from a recent paper that uses summed radiocarbon date distributions. And so this is for the Great Lakes region, and it's just looking at mastodon, mammoth, and humans. So it's a lot to look at, but just real quick. So this purple line, this is the null model of population growth, and the gray is the summed radiocarbon dates for each taxon, which is a proxy for their population. The red carpet here shows when there's a significant decline in population. And so for mastodon and mammoth, once there's a decline, there's no more population. So that's when they go extinct. And so the authors here found two interesting patterns. The first is that the populations of mammoth, mastodon, and humans are all positively correlated. So when human populations increase, mammoth and mastodon populations also increase, which is the opposite of what we would expect with overkill. With overkill, we would expect as human population increases, the population of the taxa that they're hunting should decrease, but that's not what we see. The other trend has to do with timing. So this orange bar here indicates Clovis times, and you'll see that mammoth and mastodon, their populations continue after Clovis time. So they make it through that period of time just fine, and it's not until the Younger Dryas that they go extinct. And the Younger Dryas is interesting because it is a period of increased seasonality, increased CO2, and a return to full glacial temperatures, which is unlike any other interglacial period in the past. So the authors concluded that, at least for mammoth and mastodon in the Great Lakes region, it seems that climate was the cause of their extinction rather than hunting. So in this case study, while my specific analysis doesn't directly link to modern conservation, it points to data and analyses that can provide that link. So going forward, this is how I will approach this research. I'll examine individual taxa at smaller geographic scales and collaborate with others who have experience with things like isotopes, ancient DNA, and radiocarbon histories. So approaches like these will better illuminate the links between megafauna, humans, and climate change, which will have applications to the conservation of the large mammals that we still have today, as it will allow us to see how they will respond to changes and what amounts of changes will likely lead to extinction before it's too late. And these studies can also inform the possibility of rewilding, which is the suggestion that the North American landscape should be repopulated by similar or even cloned versions of extinct taxa. However, this idea of rewilding relies on the assumption that humans were the main cause of megafauna extinctions, which at this case, or at this point, does not seem to be supported. So more solid evidence of what human or environmental factors cause these extinctions will be needed before rewilding should be seriously considered. Now, in this next case study, human and environmental factors have also been implicated in the decline of a species. But this species, at least for now, is still living, but its populations are rapidly declining. And so the drive to figure out what factors affect its well-being is perhaps more urgent. And that species is the greater sage-grouse. So the conservation of greater sage-grouse and the sagebrush habitat that they inhabit is one of the most prominent and controversial wildlife issues in Western North America today. Since the mid-20th century, the abundance and distributional range of greater sage-grouse has declined dramatically in conjunction with the reduction of suitable sagebrush habitats as a result of anthropogenic impacts and climate change. These declines have resulted in considerations of their listing under the Endangered Species Act and led to multiple multi-million-dollar broad-scale state and federally sponsored management plans. However, these management decisions are being based exclusively on this historic data. And this is problematic because even by the beginning of this record, humans have significantly disturbed sage-grouse populations, and this record only represents a relatively short period that does not encompass a lot of environmental variation. So the historic record doesn't capture the full range of range contractions and expansions that this species has experienced. Now, a species' range is not fixed. It changes through time based on a variety of factors. And in ecology, this is based on the concept of fundamental versus realized niche. So the fundamental niche is the entire range of conditions such as temperature, moisture, vegetation that an organism can tolerate and live in, whereas the realized niche is the range of conditions that an organism actually lives in at any one time. And the realized niche is usually smaller than the fundamental niche due to a range of factors such as predation and competition, geography, even human impacts. And so the realized niche can change through time based on changes in these variables. So the modern and historic record of sage-grouse only give us a snapshot of their realized niche, which has been largely shaped in historic times by human activity. So in order to conserve sage-grouse populations that we already have and successfully reintroduce populations in other areas, we need to have a better idea of their full fundamental niche. And short of extensive trial and error, the only way to do that is to obtain a longer environmental history of greater sage-grouse. And so I turned to a site that extends the sage-grouse record back 13,000 years, and that's Homestead Cave. So Homestead Cave is located in the northeastern corner of the Great Basin within the Bonneville Basin, several kilometers west of Great Salt Lake. So in the early 90s, Dave Madsen and colleagues excavated a 1 meter by 1 meter test column and unearthed what turned out to be a vast collection of small animal bones, including fish, small mammals, reptiles and amphibians, and birds that seem to have been deposited by large raptors, mainly large owls, roosting in the cave. So the Homestead Cave assemblage represents one of the most fine-grained and well-dated records of small vertebrates in North America. Not only does it contain a very local fauna because it's limited by the fairly small foraging radius of raptors, but it is also a long and continuous record spanning 13,000 years, marked by dramatic environmental change in the Bonneville Basin. So a wide range of other biotic materials from Homestead Cave, including plant materials and other fauna, have been analyzed and reported, and the trends detected in these data, paired with local paleoenvironmental data and even paleoclimatic models, give us a good idea of environmental change in the Bonneville Basin throughout this time. Although greater sage-grouse do not occur in the area of Homestead Cave today, they do occur throughout the Homestead Cave record. So the Homestead Cave assemblage is able to provide a valuable long-term local record of greater sage-grouse that can be linked to the well-known environmental history of the Bonneville Basin. So looking at things like what is the relationship between sage-grouse and various environmental conditions? What factors push sage-grouse out of an area and what factors are needed for them to recolonize an area? This information would allow us to improve management strategies for this species and allow us to identify areas conducive to successful reintroduction efforts. So I want to demonstrate this with data from the first 5 strata of Homestead Cave, or well, I guess 6, because stratum 1 was subdivided into 1A and 1B. And so these span almost 6,000 years from the beginning of this assemblage, 13,100 years ago, through the early Holocene to about 7,500 years ago. And this period covers some of the most conspicuous environmental changes in the Bonneville Basin's history. So one of the first things I wanted to look at was sagebrush, because as their name implies, sage-grouse are tightly linked to sagebrush as it provides shelter and makes up a large portion of their diet, especially in winter. So using the small mammal and plant records that have been identified from the cave, we know that at the time that these bottom strata were deposited, there were thick stands of big sagebrush near Homestead Cave. Going into the time of stratum 2, it got a little bit drier and big sagebrush stands were mixed with xeric shrubs such as shadscale, greasewood, and horsebush. And then by the time of stratum 4, it was warm and dry in the basin, and sagebrush stands were largely replaced by those xeric shrubs. So I would expect sage-grouse should follow roughly the same pattern as sagebrush. So they should be abundant early on in the record, maybe decrease a little bit in the middle here, but there should be a dramatic decline, if not disappearance, of sage-grouse by the time we get to stratum 4. And that is what we see. So this is just a plot of the relative abundance of sage-grouse, which is just the number of sage-grouse specimens divided by the total number of birds in each stratum. And so at the beginning of the record, they're present. There is this increase here at stratum 1B, which is likely linked to, it seems like there were some limber pine woodlands at the beginning around stratum 1A, and then they declined, which may have led to an increase in sagebrush habitat. So that's what I think is going on there. But they remain stable throughout stratum 2 and stratum 3, and then there's this marked decline into stratum 4, and it stays low in stratum 5. So this fits pretty well with what I predicted, and it confirms that sage-grouse were tightly tied to sagebrush habitats as in the past as they are now. But what I do want to point out is that sagebrush and sage-grouse do not always co-occur. So the map on the right here is the distribution of big sagebrush, which is the species most commonly associated with sage-grouse. And you can see that compared to the greater sage-grouse range, it's not a perfect fit. So there are areas where there's sagebrush but no sage grouse. And we have learned from failed reintroduction efforts that the presence of sagebrush habitat alone does not necessarily guarantee that sage-grouse populations will become established and survive. So while sagebrush plays an important role, it seems that there are likely other climatic factors that shape sage-grouse's niche. So to investigate this, I utilized the macrophysical climate model that Broughton et al. 2008 created for Lakeside, which is right near Homestead Cave. So briefly, macrophysical climate models, or MCMs, are similar to general circulation models, which use things like sea surface temperature, insolation, CO2 concentrations to simulate past climate. The difference is MCMs also use data from historic weather stations in order to calibrate the model to a local area. So this particular model is specifically tailored to the area of Homestead Cave. So here's an example from the model. This is the model temperature range. And so the model provides averages at 200-year intervals. And this MCM models a range of climatic variables that have been found to affect vertebrate taxa, like precipitation seasonality, temperature seasonality, and overall moisture. So I was interested in what modeled values of these different variables looked like during the section of Homestead Cave that I'm looking at. So what was the climate like when sage-grouse were abundant, and what was it like when they declined? Is there a difference there? So I examined 3 of the climatic variables produced by the MCM: moisture index, temperature range, and summer precipitation. And I thought that these were likely to have an effect on sage-grouse populations. So the moisture index is a measure of annual effective moisture. So it's the annual measure of precipitation weighted by water surplus and water loss. So it can show periods of droughts, which I think would have a negative effect on sage-grouse. Temperature range is a measure of temperature seasonality. So all it is the difference in extreme winter lows and extreme summer highs. So high temperature ranges indicate really hot summers and really intense winters, which I think are also likely to negatively affect sage-grouse populations. And then summer precipitation is just the percentage of the total annual precipitation that falls during July and August. And so summer precipitation can affect insects and small buds and other high quality forage that sage-grouse and their little chicks, pictured here, rely on during the summer months. Okay, so this plot shows these 3 variables. So the moisture index on this axis, temperature range on this axis, and summer precipitation on this axis. And so each of these dots represent the value of each of these 3 climatic variables during the time that that stratum was deposited. So you can see that the strata where sage-grouse were abundant, so 1A to 3, kind of cluster together, and the 2 where sage-grouse declined kind of cluster together. And so all of this space that contain these 6 points, all of this represents the sage-grouse's fundamental niche, right? Because even though they were there in low numbers during strata 4 and 5, they were still there. They could still exist there. So this is all their fundamental niche, but it seems like this area that contains these 4 points is where sage-grouse do better. And so it's kind of a mid-range to higher moisture index, a little bit higher temperature range, and higher summer precipitation. So once I add in the entire homestead record, I think it'll give us a clearer picture of their preferred climate. And then the next step would be projecting that onto a modern map so that we'll be able to see climatic conditions that look like this, that are not conducive to sage-grouse. If there are sage-grouse populations living there, we know those are the ones we'll have to keep an eye on. And then we can identify areas on the map that have climatic variables in this area that might be conducive to successful sage-grouse reintroduction. And then I'm also working on assessing the potential effect of human hunting on sage-grouse populations. So there are 2 archaeological sites in the area that are well dated and that contain sage-grouse remains, Danger Cave and Bonneville Estates Rock Shelter. And the relative abundance of sage-grouse in these assemblages roughly mirrors that of Homestead Cave. So they're abundant early on in the record and then decline into the early Holocene. Right now, these records show that people were hunting sage-grouse when they were around, which makes sense. They're big, tasty birds. But whether or not this hunting depressed sage-grouse populations or otherwise negatively affected them remains to be seen. But from the data that I presented today, it's clear that sage-grouse are fundamentally linked to sagebrush, but they are also affected by other climatic conditions, such as moisture, seasonal precipitation, and temperature seasonality. So understanding the full range of conditions that sage-grouse can tolerate, so their fundamental niche, and the conditions under which they thrive could help wildlife managers predict how populations will respond to climate change and identify populations that may be most vulnerable, and most importantly, can help decide where sage-grouse can be successfully reintroduced. So knowing this information also opens up the possibility of establishing successful populations outside of their historic range, which is important because a lot of their historic range right now is too anthropogenically disturbed or unavailable. And so establishing populations outside of their historic range may be crucial to the survival of the species. And Homestead Cave may be the key to that. But this is not just limited to sage-grouse. So Homestead Cave contains records of at least 60 different bird species, including many regionally important species like waterfowl and shorebirds, whose continued existence is crucial to the nearby Great Salt Lake environment. And it contains other taxa that have received a lot of conservation attention locally, such as the snowy plover and the yellow-billed cuckoo. And so this is extremely valuable for modern bird conservation because as you've seen, it greatly extends the record that we have for many of these taxa. So I'll be applying for an NSF grant next year in order to continue this work with the Homestead collection and begin to expand out to other nearby areas, like possibly the Snake River Basin, to investigate if these local trends that I'm seeing in Homestead are also consistent regionally. And this huge data set also offers ample opportunity for student projects. And I'm willing to mentor undergraduates or graduate students and help them come up with their own research questions that can be addressed with this data set. So as I, or potentially students as well, continue this work with the Homestead assemblage, it's important to get these data into the hands of people actually making policies and decisions about bird conservation. And so I'm already in contact with the Washington Department of Fish and Wildlife about using my data for their sage-grouse conservation strategies. And I've been invited to speak at the Sage and Columbian Sharp-tailed Grouse Conference this summer, which is hosted by the Western Association of Fish and Wildlife Agencies. Next month, I'm also giving a talk to the Great Salt Lake technical team, where I'll be focusing more on the conservation of aquatic birds that utilize Great Salt Lake. So this sort of collaboration is what will help make my research actually relevant and useful to conservation. So I'll be continuing to seek out more connections within the wildlife and land management communities. So these 2 case studies of my work examining the past relationships between animals, the environment, and humans exemplify the variety of contexts within which this approach can be used. And unfortunately, this kind of work is the need for it is increasing. Many species are already in peril or headed that way, and climate change and human impacts on the environment are only going to increase as well. So as a result, we're likely to see more extreme conditions that we've never seen before in historic times, but that likely have analogs in the past. So being able to look back at the records of the past in this way will be the key to conserving the future. So before I end, I just want to say a quick thank you to the Natural History Museum of Utah for funding and access to collections, especially Glenn and Nielsen Grimm at the museum. I also want to thank the University of Utah Archaeological Center and my collaborators and mentors, Jack Broughton, Brian Cotting, Tyler Faith, Mike Cannon, and Joan Coltrane. And so with that, I think we have a few minutes left if anyone has any questions. I'm not sure if you can open up the chat. Okay, let's see. Maybe.
Hello, this is Pei Lin. Can you hear me?
Yes, I can hear you.
Hi, I think there's a couple of questions, including a couple from me in the chat box.
Okay, yeah. Yeah, if someone could just read them, that might be easiest.
I'm happy to read mine, if that's okay.
Yeah, go for it.
Or sort of rephrase it. I was, sort of asking a couple questions about the earlier part of your talk, and congratulations on a very, very interesting talk. Regarding the notion of encounter, you were talking about, the various aspects of that, and I was wondering if there's a way to sort out different kinds of encounters. For example, an encounter with a healthy adult animal versus an encounter with a dying animal, or scavenging from a kill that's been made by another predator. It seems like the way that encounters play out could really shift the rank of the prey up and down.
Right. Yeah, that's a very valid point. At this point, I'm not aware of any way to do that archaeologically, and that's kind of the problem you mentioned about like scavenging like sick or dying animals. That unfortunately is something that we have yet to figure out how to discriminate in the archaeological record. So these associations, the 15 sites that we have that have been scrutinized, the associations include things like artifacts very tightly, unquestionably linked to remains of megafauna. It includes things like cut marks, sometimes even projectile points embedded within bones. But from that, you can't tell if the animal was hunted. You can't tell if it was just scavenged, although I guess projectile points being embedded wouldn't suggest more hunting, but those are very, very rare. But so that's why it's archaeological kill / scavenging sites. There's not a great way, at least that I know of, to discriminate between those two. And I think that, like you mentioned, would have an effect on encounter rates. Like a question that I get fairly often is people don't believe that human hunters were hunting taxa like mammoths and mastodons because they may have been really dangerous to attempt to hunt. But if you come across one that is old or dying or already dead from another predator, it's still a big package of meat. So you'd still want to cut some pieces off and take it with you. But the archaeological remains of that don't tell you much about whether that was the case or whether it was actually hunted. So it is really rough data in that way.
And I know that the sample size is really small too, but I'm thinking about a great paper that Pat Shipman wrote back, I think in the 1980s, about cut mark stratigraphy and tooth mark stratigraphy. And I think that this has been done in East African assemblages where, folks are trying to sort out when the Australopithecines shifted from scavenging toward hunting, and where you actually can take a look, if you have the data, as to whether cut marks underlie predator tooth marks or scavenger tooth marks, or whether they actually cut through those tooth marks. It's a really cool paper. I mean, there might be something in the African literature that might be helpful.
Yeah, and even like what is considered secure archaeological evidence is still debated in this context. The paper that I showed, Grayson and Meltzer's 2015 paper where I showed their graph, that paper, the point of that was actually re-examining some evidence of these archaeological associations between extinct taxa and humans. And so that's where that 15 number came from. So they had a set of really stringent criteria that they went through, and it was an update from an earlier paper where they added some sites, they took out some sites. So that is, can be subjective in a way where a lot of people may argue that there are a lot more sites than that, based on cut mark evidence that may or may not be legitimate. Anyway, my point is that there's no real consensus on that, no wide consensus on that.
Thank you very much. And I see there's a question from Eric Robinson too there. I don't know, Eric, if you want to read that one.
Yeah, I can just, I don't know if it came out clearly. So great talk, Allison. Thanks a lot. Can you hear me?
Yes, I can hear you.
Okay. So great talk. Thanks a lot. I found it really interesting that in your map, you keep showing about the differences in the historic and the current sage-grouse habitats. It seems that in the north, there's a definite difference in the northern part of the distribution of that map than there is the south, meaning that there is a greater overlap in the southern regions between the historic and the current habitats. I'm wondering if you could elaborate a little more on what you've learned from your work, because you're wanting to apply these archaeological records to modern conservation issues. So I'm just really curious if you could elaborate more on why those differences exist. If you know why they exist, is it an agricultural, sort of industrial agriculture thing in Montana? I'm just kind of curious about what that can shed in terms of like the human behavioral decisions to cause those differences. Does that make sense?
I think I know what you're getting at. Let me see. You can let me know if I answered it or not. So I think the differences in like the different geographic areas of where, let me see if so in the north, there's a difference than in the south of the overlap between sagebrush and sage-grouse. Is that what you're pointing out?
Yeah, there seems to be much more of an overlap in areas like Wyoming, southwest Idaho, southeast Oregon, Nevada.
Yeah, so that I think has to do with a range of factors. A lot of these places are more developed than others. For example, there's likely a lot more development in Idaho than there is in Wyoming. There's a lot more open space in Wyoming. And there have been a lot of efforts to restore sagebrush habitat in the West. So not necessarily associated with sage-grouse themselves, but with restoring sagebrush habitat. A lot of the ranchers are getting on board with land management teams and trying to figure out how to restore that habitat. But it differs by state and it differs by region. So at this point, like I don't have a great answer for you for that, but that's part of the reason why I want to team up with wildlife managers and land managers, because I'm not a wildlife biologist. I don't know the ins and outs of a lot of this, but what I do bring to the table is this data set, this really long data set. So being able to work with people who are on the ground who would probably know the answer to your question, I think is key. So to move my data out from academia and put it into a place where it can actually be useful for people who know what they're doing with sage-grouse conservation, that could answer questions like this.
Okay, and a follow-up is the chronology of Homestead Cave is kind of interesting, and I'm just curious. A lot of the levels are occurring around periods of very, very interesting centennial scale, multi-centennial scale climate change events.
Yeah.
So I'm just curious if you could elaborate on how that assemblage might be helpful. Do you have evidence of responses of faunal communities there to abrupt climate change events, things like that? Because I mean, it's capturing some really interesting changes at the beginning of the Holocene, especially.
Yeah, so that's something that I'm working on once I have the full homestead record. But there have been studies with other taxa. I know Jack Broughton did a study on the artiodactyls from the area, and the fish as well. The small mammals have been extensively studied by Grayson and Terry and Novak. And yeah, it seems that a lot of these taxa are tracking some of those centennial scale events. And like, I mean, the most obvious one is at the very beginning of the Homestead record. It captures the final regression of Lake Bonneville. So right at the beginning of the Homestead record is when Lake Pleistocene, when the lake level dropped to modern day Great Salt Lake levels. And so that was a huge change in the Bonneville Basin. And you can't really see much of that with the sage grouse, but this is what I'm working on with some of the other aquatic taxa that I'll be presenting to the Great Salt Lake technical team, where you can really see that shift in things like diving ducks versus dabbling ducks, or like big piscivorous grebes versus more shallow water grebes. So this Homestead record is really cool in that we have so much material, so many different animal taxa that we really can answer questions like that. And they do seem to be tracking those big climatic events really well.
Great, thank you so much.
Okay, I still haven't figured out how to see the chat. Wait, I think I can, oh, here we go, let's see. Oh, no. Oh, here we go. I can either read it or yeah, sorry.
There's one more that came through that says, in terms of the proportions of the Homestead and ISPs, differences were in the few 1/100th% range. What values of N do these reflect? And Chromebook Theo 6F can add to that if they'd like.
Yeah, so talking about this, the reason that they're so small is because there's a huge number of bird remains in each strata. So for instance, like in stratum 5, stratum 5 is 0, but stratum 4, I think it's just like a handful of sage grouse remains. I have played around with this to see if it makes a difference if I use different indices. So sage grouse specimens divided by the total number of just terrestrial birds, or divided by the number of like grouse-like taxa, which there aren't too many other besides sage grouse during this period. But it all reflects the same trend. So I just defaulted back to using total number of specimens, of bird specimens for this. But this pattern does hold with no matter how I measure it.
Follow up to that is, have you tried to bootstrap any error estimates for these?
I have not. That would be something that I would look at more closely once I have the entire record. But it seems like, I mean, just from, like I said, just from the trends seen in other like mammalian taxa and fish taxa, I think it should still hold.
Great, thank you.
Okay, is that it from the questions in the chat? Okay.
Yeah, that, as of right now, that's all there is.
Okay, thank you. Any other questions from anyone else?
You had briefly talked about looking at other bird species in addition to sage grouse in the records. I was just kind of curious to know what your thoughts on how those would be applied, and talking about applying your research to a broader context of management. And just kind of thoughts on what you were thinking with.
Yeah, so like I mentioned, I'm working a lot with the aquatic birds, so the waterfowl and the grebes and the other shore birds. Great Salt Lake has a really unique avifaunal assemblage today. It's really important for a lot of species, a lot of migrating taxa. It provides a stopover place. A lot of bird taxa winter there. It's one of the premier bird watching sites in Western North America. So a lot of what we see today it was not always like that. And so one of my big questions I'm addressing besides sage grouse is looking at the changes in those aquatic taxa, because there are a lot of efforts now to manage Great Salt Lake as they say mostly a waterfowl management area, but they really mean all of the other aquatic taxa. And so I've approached them, and like I said, I'm talking to them next month to see what they think about collaborating, to see if my data that I have, this really long record, if that would be useful to them. And then, like I mentioned too, there's a couple other species that are really being pushed for management in Utah, like the yellow-billed cuckoo is one of them. And I've happened to find yellow-billed cuckoo in the homestead assemblage. I haven't done anything with that yet, but that's definitely next on my list. Sagegrouse is a big hot button issue, and it's really time sensitive right now. Yellow-billed cuckoo are doing a little bit better, so I prioritize sagegrouse, but that's definitely next on my list as well.
That's
okay,
awesome. Thank you so much.
We had a question in person here. The sagebrush, oh, I guess I didn't put a citation. Oh, you mean when I showed this, wish this clicked faster, this. Okay, yeah, so that, it's not quantitative. There are quantitative ways to do this. There have been pollen records done in the Bonneville Basin, so that I want to definitely corroborate this with the pollen records, but this in general is just based off of trends that are from the other materials that have been analyzed from Homestead Cave. So there's plant macrofossils, there's pack rat middens in the cave that have a lot of plant macrofossils as well. And the small mammal record has informed on vegetation changes in the area. There are a lot of other sage obligate mammalian taxa. So those two lines of evidence, the plant macrofossils and the small mammal record, are what I'm basing this off of, that there's likely sagebrush early on and then it declines into Shriam 5. All right, I guess that's it for questions. Thank you.